I'm going to talk about the plasma membrane, which is the selective boundary on the outside of all cells. Some cells, eukaryotic cells, also have membranes inside their cells, like mitochondria surrounded by membranes, lysosomes are surrounded by membranes, the Golgi is surrounded by membranes. It doesn't matter if you know these names yet, but these are all membrane compartments. So all the stuff I'm going to talk about with the plasma membrane, or the membrane on the outside of the cell, also applies to how membranes behave in these compartments. So the compartment membranes are the same thing. They are composed, all these membranes, of lipids that are specifically called phospholipids, and that's because they're made of a phosphate group. It's a group of phosphorus and some oxygens. It's negatively charged. And nonpolar tails, which are these long chains of carbon with hydrogens attached. Much longer than this, etc. So these carbon-hydrogen bonds are nonpolar, meaning their electrons are distributed evenly, meaning they have no charge. So we have a negatively charged phosphate side of the phospholipid, and we have a no charge tail side of the phospholipid. And so you can see that there are hydrophilic and hydrophobic. So hydrophobic goes with nonpolar, and all that means is that it does not mix well with water. So this core here, the tails, doesn't mix well with water. That's hydrophobic. Phobic means fear. Hydro is water. They're not actually fearing water, but they turn away from it because they don't mix well. Hydrophilic, again, hydro is water. Philic or phile means lover. So these are water lovers. Then those negative charges on the phosphate groups interact well with water. They can bond with it. And water is on the outside of the cell, on this side, and water is on the inside of the cell, down here. So these negative phosphates face the outside and the inside of the cell, whereas these tails are sandwiched in between. This matters because this bilayer, 2 layers of phospholipids, you can see that here, 1 layer, 2 layers, and here's the in-between. That bilayer is selective, meaning that it only lets certain things through. So certain molecules can go through the tails. Other molecules will not be able to go through the tails and will stay outside the cell or inside the cell. And that's part of transport and how that works. The selectivity is due to this hydrophobic tails. Those hydrophobic tails are what gives it selectivity. The heads just face the outsides. They don't dictate what goes through or not. It's that core, the sandwich. And remember, these are cut-throughs of membranes. Membranes are really a sac, like totally closed in, where the heads are both on the outside facing the exterior, and they're on the entire inside, and then the tails are sandwiched in between. So the heads are lining both the outside and the inside with the tail sandwich. A second part of the membrane is cholesterol. That's these blue structures in here, the teal. They are made of, they are also lipids, specifically a steroid type lipid. And they're kind of rigid, but not too rigid. They are made of fused rings, hexagon, you don't need to know the specific shape. But there we go. So it's something like this with rings. And so it is rigid which means it keeps the membrane a little bit stiff. But it also spaces the phospholipids away from each other, which also keeps it sort of fluid. P-lipids are phospholipids. So in the end, this ring structure of cholesterol regulates the fluidity, meaning how much the phospholipids move around. So if it's stiff or rigid, it means they don't move around very much. If it's fluid, they're moving within their layer, but they're moving around a lot, and so cholesterol helps regulate that. Cells can actually change the amount of cholesterol they have to make their membranes more or less rigid, or parts of the membrane more or less rigid. So that's something we see is places that have a lot of cholesterol get stiffer and other places don't. There are also embedded proteins, so that's these big purple structures. They differ from cell to cell, and that dictates a lot of the cell function. So which proteins they have in the membranes allows them to do different things. So this one here in the front left looks like a transport channel to me. So it looks like it probably lets molecules in or out of the cell. Now, if another cell didn't have that same protein, it wouldn't be able to do that. Maybe this is some sort of signaling molecule that can receive a signal in from the environment and transfer the information to the inside. Other cells that don't have that signaling molecule couldn't do that. So the proteins help the cells be specific. Glycoproteins, which is what you see here, are proteins, this purple part, plus this green part here, which is a carbohydrate chain. That's the glyco, is the carb part, and then the protein. So it has these medium chain carbohydrates, and they're important because they identify cells. So different cells have different carbohydrate chains, different types. So it's either different sugars or different lengths or both. And those are identifiers for the cell. So for example, your blood type, your ABO blood type, A's have A tags, B's have B tags, O's have no tags. We also have MN blood type. So some people have the M tag and some people have the N tag. You have different sorts of immune, the MHC complexes. These complexes identify and they stick out and identify different molecules in the immune system. And they can detect whether other cells are self cells, meaning your own cells, or foreign invaders based off of these tags. So this is important because your cells should only recognize foreign cells as foreign. But if they accidentally have some problems and they recognize your body cells as foreign because they don't recognize the tags, then you end up with autoimmune diseases where your body is attacking itself. So those are some parts of the plasma membrane and their functions to give the membrane its characteristics.